Hello, so this is 11.4 Sexual Reproduction, IB Bio. So your learning objectives, compare and contrast spermatogenesis, so we'll find out what that is, and oogenesis, and we're going to evaluate some advantages, disadvantages of internal and external fertilization, and explain the process of gestation and birth, which is quite interesting. And then your homework is to discuss the impact of birth control pills on the environment. I'm in the library right now, so apologies if it's a bit noisy. We'll be back on track later. So spermatogenesis, this is something where we're creating sperm. So what you've got is something called spermatogonium. Now these are like your stem cells, and they're going to undergo mitosis. So we're over here at the top of this diagram on the right. When the spermatogonium undergo mitosis, what's happening is one of the cells that are produced, because remember mitosis, you get two identical daughter cells usually. One of them will become something called a primary spermatocyte, while the other one will become another spermatogonium cell. So that means you maintain the number of these sperm stem cells in your testes, and that's how we continue to make sperm in testes. So what happens is the primary spermatocyte undergoes meiosis 1. Now in meiosis 1, what happens is the chromosome number halves. So you now have, it goes from diploid in the primary spermatocyte to a haploid number of cells. Then what happens is the primary spermatocyte undergoes meiosis 2, and then that you get something called spermatids. So now you've got from one spermatogonium stem cell, you have 4 spermatids. And then the 4 spermatids, what they'll do is they'll travel down to something called Sertoli cells. We're on step 4. And the Sertoli cells will nurse the spermatids with nutrients until they become sperm cells, the typical sperm cell that you've really learned in year 7. And that's over here, these cells. So what's so we also need to be able to annotate the diagram in the middle, which is here. So you need to know what's happening here. So we start off at this level, which is the spermatogonium cells. These are stem cells. Then we've got the mitotic division to make the spermatocytes. That's in this section here. And then in some diagrams, they'll show like some sperm cells undergoing mitosis and the chromosomes going from one place, like anaphase and metaphase and so on. And then they also show meiosis 1 and 2 and 3 and things like this. No, there's no meiosis 3. And then you've got the secondary spermatocytes, which are down here. You've got Sertoli cells, which are these cells. Can't really see them anywhere actually, but they're around here. And I'll show you on the next diagram anyway. And then you finally got the sperm cells here. Sometimes the diagrams flip this way or sideways or the other way, so just watch out for that. You also might hear of things called Leydig cells. So Leydig cells are just basically the other kind of cells that are found in between the rest of the testes, I guess, is the interstitial tissue. So moving on. You also need to be able to annotate a diagram of this sperm cell. So this is a diagram of a mature sperm. You know the general shape. So you've got the head in a pointy position. At the top of the head is the acrosome. That contains hydrolytic enzymes, and we're going to find out what that's used for, but you probably know it's used for digesting through the plasma membrane of an egg cell, but we're going to find out in more detail. A haploid nucleus, so half the number of chromosomes. You have something called the midpiece, and inside it is helical mitochondria. So that's pretty cool, and you can look more into that later, but yeah, that provides ATP to move the tail. Okay. So here is a bit more detail. The diagram's now flipped. So at the bottom, here's your interstitial cells that I was talking about. Then you've got your spermatogonium. So these are your stem cells, and they undergo mitosis to make primary spermatocytes. And the primary spermatocytes then become secondary spermatocytes. They become spermatids, and the spermatids then differentiate into sperm. And these things, and this here is your Sertoli cell. So you can't really see because I haven't done a good diagram. but your totally cells would be here like this, and the sperm cells would be nursing themselves into, or the spermatotids will be nursing themselves into sperm cells at these Sertoli cells. So these are the Sertoli cells here. Okay, so try this. So I'll give you some time. I think you need to pause this video because I'm not going to pause it. So describe the process of spermatogenesis leading to the production of 4 sperm cells in a human male. So you need to think what keywords you're going to hit with that one. Maybe rewind back, think about the keywords. And then for number 2, I can't move myself out of the way. Wait, I can. Okay, I can't move myself out of the way, unfortunately. So just try which one of those. So which labeled cells are diploid and haploid respectively. So have a go with that. Or you can just go through each one and think which one's haploid and diploid. And then yeah. So pause it, have a go, and I'll go through the answers in 5, 4, 3, 2, 1. Okay, so for number 1, the answers are here. So if you said seminiferous tubule, you get a mark. Diploid cells, Spartagonia, grow, you get a mark. 2 divisions of meiosis, if you mention that through the list of things, you get a mark. I think there's 4 marks for question 1, sorry. Primary spermatocyte carries out the first division, and so on. So the rest is there. And for number 2, W is going to be diploid. X is a Sertoli cell, so that's also diploid. You can see because the sperm cells are nursing off this one big cell. So this is that long big Sertoli cell that the other diagrams fail to show. Y is haploid, that's a sperm cell, or spermatozonia, and Z is a secondary spermatocyte. Oh, sorry, these are spermatids, beg your pardon. So Z spermatids. And the answer was C. So oogenesis is similar to spermatogenesis, but in oogenesis, you're producing egg cells. So this is quite cool. So when very early on in fetal development, the number of eggs inside the female fetus is about 1 million, maybe. Then at birth, the number of eggs goes down to about 300 to 400,000. And then by the time you get to, that number reduces by the time you get to puberty, and then the number continues to reduce more than the number of eggs that are released during menstruation, until menopause, where no more eggs are produced, and no more menstruation occurs with the female. So anyway, going to here, so oogonium is produced by mitosis. This is happening in the embryo during fetal development. So we're going from the top here. So you've got an oogonium, you get 1 mitotic division, okay, and the number is still, the chromosome number is still diploid, and the primary oocyte is arrested in something called prophase of meiosis 1. So it's prophase 1. Okay, so you've got primary oocyte. Then, when you turn anywhere between 9 years old to 13 years old, puberty occurs, and every month the levels of FSH will increase, and that will cause the the initiation of the first mitotic division. When the first mitotic division occurs, then you have a secondary oocyte, and that is arrested at metaphase of meiosis 2. So it's arrested in metaphase 2. Okay, so this is where you've got the centrioles moving, you've got spindle fibers almost being formed, almost going to touch the kinetochores of the centromeres of the chromosomes, all this sort of things that you learn in Unit 3 and you might learn later in Unit 10. So once a secondary oocyte is formed, then what happens is we have to wait until fertilization, and after fertilization, the rest of meiosis 2 will continue, and we'll look into that later. What's happening though in the first meiosis and the second meiosis is you have something called the production of a polar body, and what happens is the polar body just degenerates. But it's quite interesting because you've got the oocytes, I guess, trapped in a follicle, and when one polar body is formed, it stays in this kind of like follicle of cells. The reason why this is so complicated compared to sperm cells is because the egg cells need so many nutrients. They need enzymes. They need they need something called cortisol to carry out something called a cortisol reaction. They need to have so many different other layers that we're going to look into later in this lesson. So that's why it's more complex than a sperm, the production of sperm. I'm going to show you a video next, actually, on oogenesis. So have a look here. Oh, I can't click it. Wait 1 second. Here we go. So have a watch of this. And that's the production there on the left of a polar body, but it stays in this follicle. So they both kind of stay in the same place. You can clearly tell which one is the primary or secondary oocyte that is formed. And you can clearly tell which one is the polar body. And what's happening is the primary or secondary oocytes keeping most of the cytoplasm, because that's where the good stuff is. It's where all the enzymes and the nutrients need to be. And if it divides equally, then they're going to both have, well, one's going to have a bit more, but the other one's going to have less. You know, your primary oocyte is going to have less of the stuff it needs. And that's why this division occurs in this way. So it's evolutionary beneficial. So yeah, moving on. So here's another diagram. So what we're starting off here is the primary follicle. Okay, so this primary follicle contains a primary oocyte, then the secondary follicle, and so on. Then you've got something called Graafian follicles, a mature follicle. Okay, and at this point here, you have a secondary oocyte that's trapped in our Graafian follicle. What's actually happening is when it comes to the Graafian follicle, you actually have a layer of follicles, probably like 3 or 4 layers of follicles, and that's kind of making this one thick, large follicle that surrounds the oocyte. So you've got the secondary oocyte at this point. Then what happens is the kind of outside of the follicle disintegrates a little bit, and you get something called a corpus luteum. So corpus means body in Latin, and luteum means kind of like dirty. So it's like a leftover part of that, and that also plays a crucial role later on. And we'll also look into what the secondary oocyte does later. What I've got for you next is I've got some questions here, multiple choice questions. So again, pause it, have a think, and in 5 seconds I'm going to go through it. 5, 4, 3, 2. Okay, we're going to go through it now. So for number 1, the answer, if it ever lets me click, is FSH. Number 2 was prophase of meiosis 1. Number 3 is metaphase of meiosis 2. And number 4 is fertilization. So what I'd like you to do now is you can use either Cognity or go to this link here. This is the direct link to BioNinja, and you need to compare and contrast the differences between spermatogenesis and oogenesis, and include information about the reproductive tissue as well, like kind of how the gamete, the production of gametes moves through. Another key word which is not on this PowerPoint is gametogenesis. So that's just the collective term for making sperm and eggs. So oogenesis, spermatogenesis, this kind of fits under the umbrella term of gametogenesis. So this is a quick one. It's just regurgitation of facts in this case. So internal versus external fertilization. Internal fertilization, as you know from the diagram of the cock and the hen, it's just internal fertilization. So the sperm goes into the vagina and fertilizes the egg in the uterus. Now, some fish and some amphibians, they do something where they spawn. So what that is they'll fertilize the organism will release eggs into water, and there's loads of eggs, and then the male would release lots of sperm in the water, and they'll meet together outside. Well, the advantage of that is that you get a large number of offspring that are more likely to survive, even though they're susceptible to predators, obviously, in the water. Whereas internal fertilization, what you have is copulation occurs, So that's sexual intercourse. You transfer the gametes inside the hen, but then you only get 1 or 2. In some cases, you get 5 or 6, like a litter of babies. But there's a larger gestation period, I guess, and then I guess there's a chance of harm to the female because the female has to carry it. So there's likelihood of, in the wild, that female is very unlikely to survive unless protected. Whereas with external fertilization, once the female or male have released whatever they need to release, egg or sperm, they go, and that's it. So yeah, those are some differences there. There's a table there as well. Moving on. So internal fertilization. So what happens is in internal fertilization, the sperm enters into the vagina and then travels up past the cervix. So the cervix is like the opening into the uterus. Uterus is where the baby is going to be. And then the sperm goes into either fallopian tube, left or right, so 50-50%. Sometimes that percentage is a bit tweaked depending on other factors. And then the sperm travels up the fallopian tube. The blastocyst, which is kind of like the unfertilized egg, kind of releases some chemicals as well, which attract the sperm. Another thing is that the sperm is surrounded, as it's traveling up the vagina, surrounded by this kind of like cholesterol layer, and it's very hard for the sperm to swim. So as it travels up, what's actually happening is that cholesterol layer is like broken down by the acidic environment in the vagina, and that helps the sperm swim faster. So yeah, by the time the sperm reaches the egg, as you know, only one sperm, the fastest, strongest sperm wins, and that's how humans and also mammals, we make, evolutionary speaking again, that's how we make sure the best of the best is always born. So the best sperm is always going to win and fertilize the egg. But we need to look into how this fertilization process occurs. So what's happening here is the sperm meets the egg. There's actually loads of cells around the egg that was not in the diagram. Loads of these kind of cells. These bunch of cells are known as the corona radiata. So the corona radiata, corona as you know means crown, radiata just means it radiates around the periphery of the egg cell. So the corona radiata, the sperm kind of burrows its way through those kind of bunches of layers of cells, and then it reaches something called the zona pellucida, and this is where the hydrolytic enzymes in the acrosome of the sperm's head kind of starts digesting away. And as it digests away, it reaches some receptors in the egg cell. And the receptors, when they feel the sperm's presence, I guess, by recepting the chemicals that are there, they cause something called cortical granules to release something, which I don't know, but they release something. And what they do is the cortical granules react with the zona pellucida and cause the zona pellucida to harden, which will prevent any other sperm from reaching the egg. And what we're trying to prevent is something called polyspermy, which is where more than one sperm meets the egg. Okay, so that's the main thing. Once the sperm has entered into the egg and gone past the plasma membrane of the egg cell, then the sperm's nucleus, which is haploid, will meet with the egg's nucleus. And at that point, when the sperm's, before that point, so when the sperm reaches the plasma membrane of the egg, then the egg cell is going to finish doing metaphase 2, because remember it's arrested in metaphase 2. It'll finish that, and one set of chromosomes will move towards the egg cell, and the other set of chromosomes will make another polar body that's trapped in this kind of area of follicle cells. But that would just degenerate later on anyway, so we don't need to worry about that. You just need to know polar body's made and then degenerates later. Next stage is implantation. So what's happening is after fertilization, which is here, okay, fusion of sperm and egg cell, yada yada yada, you get a zygote. So the next stage is zygote. And then after a number of days, what's happening is you get something called, so we'll skip all of this, 2 cells become 4, 4, 8, you get a bunch of cells, a morula is formed, which is like a bunch of just cells that have undergone mitosis from the zygote. Then you get something called a blastocyst, and the blastocyst is where we need to focus on. So the blastocyst is quite interesting because it's made up at this point between the morula and the blastocyst. The cell division is not genetically, well, it is genetically identical, but they're not. If you look at the cells, they look a bit different. So some cells are like long and flat, some cells are like normal ball shaped, some cells as like one large cell with nothing in it. Okay, so there's lots of different cells going on here. So what happens is when this blastocyst is implanted into the endometrium lining, that releases something called HCG, which is human chorionic hormone. Now, human chorionic hormone is the thing that's detected in the urine test when women need to test if they're pregnant, which occurs around 2 weeks of after the latest menstruation. So what this does, this HCG, is it prevents the degradation of something called the corpus luteum, which we learned about earlier, that dead body that occurs in the ovary after the egg's been released through ovulation. What the corpus luteum does is it releases progesterone and estrogen, and these continue thickening the endometrium lining and prevents menstruation from occurring, because menstruation is the release of the egg through the, when you have a period, the release of the egg and also the release of the lining, which is why it's blood that comes out, because it's the uterus lining that's coming out. So that's what progesterone and estrogen are doing. Okay. What also happens is, in this area where the implantation is occurring, is that HCG promotes the growth of the placenta, which we'll look into next slide. So let's look into that now. So here is the placenta. So the placenta is basically just an organ that grows specifically for gestation, which is when the baby's growing, the fetus is growing. And what the placenta allows is the exchange of materials between the fetus and the mother. So, the mother's going to give the fetus oxygen, going to give all the nutrients, amino acid, fats, sugar, carbs, antibodies as well, so that the immune system, parts of the immune system are shared, all the vitamins, water. Unfortunately, also if the woman smokes or has alcohol or has any drug abuse, that unfortunately is also transferred to the baby through the blood. So that's the placenta is where it's happening. The umbilical cord is the part that connects the placenta to the baby. And if you zoom in here to this diagram, you can see that clearly, this diagram here. So you can see the umbilical cord here, and you have these villi here, which increase the surface area, so you get a better rate of absorption. I'm saying absorption because it's not just diffusion. You have endocytosis, exocytosis, facilitated transport, facilitated diffusion as well. So you've got more than just one type of diffusion occurring. I'm going to look at that next. The fetus, in return, because it's so grateful, is going to give back CO2, because obviously the fetus is respiring. It's going to give back urea, because the fetus is producing waste from breaking down amino acids and other substances. It's also going to give back hormones, which is why women who are pregnant experience changes in hormones, and so on. Another thing is that the placenta causes an increase in estrogen and progesterone concentrations, which then furthers this development of the lining as well. Here is a quick little table on what's transferred, so you can pause it and make notes from here. So what's transferred from the mother to the fetus, what processes are involved, and then also what is passed from the fetus back to the mother and the process which each one does. So I'll let you pause and take notes on that. But you need to know this, that's what I put it on. Okay, and then finally, not finally, we've still got 2 more slides. But so at birth, what happens is the fetus signals the placenta to stop producing progesterone. This then causes oxytocin to be released from the mother's pituitary gland, the posterior pituitary. It also from the baby's posterior pituitary. So those that do psychology know that oxytocin has a role in happiness as well. But in this case, it's more to do with birth. So what the oxytocin does is it starts causing muscle contractions. What also happens is, as I said earlier, the fetus signals placenta to stop producing progesterone. What also happens is the estrogen continues to be released from the placenta, causing increase in receptors of the uterine muscles. So yes, you get more oxytocin being released, but then you get more oxytocin receptors being produced in the uterine muscles of the pregnant female. So yeah, that's what happens. And then the dilation of the cervix results in a positive feedback loop. So what happens is, as the cervix dilates, as the baby's head pushes against the cervix, so this is the cervix, this is the baby's head, pushes against the cervix starts opening a bit, okay, then that causes more oxytocin to be released, which causes more contraction. And then when it gets to a certain stage, well, I suppose when the contraction starts, this is known as being in labor. And part of that is when the water bursts. What's happening there is the amniotic fluid, which is surrounding the baby, which is the thing that causes fingerprints, by the way, in the fetus, in the amniotic sac. What's happening is, as the baby's head is pushed against the cervix, that causes the amniotic sac to burst and all the fluid coming out. So that's when your water breaks. So that's pretty much birth in a nutshell. Now we're going to quickly look at gestation versus lifespan and gestation versus size as well. So typically, as the size of the mammal increases, there seems to be a positive correlation with that and the gestation period, so how long the mammal holds the baby in its uterus. But that's more, it could be causation rather than correlation. So the other factor involved in this is that typically mammals that are more complex when they are born tend to have a longer gestation period than mammals that are not as complex. So you've got two types of mammals that are born. You've got altricial or altricial mammals, and you have precochial mammals. So altricial are those that are helpless, so humans are born helpless, and precochial are independent, so things like horses, cows, they come out and they start walking, giraffes, and so on. So the more precochial mammals, they tend to have a shorter gestation period than the altricial babies, infants. Okay, so your homework is looking at estrogen pollution. So you need to complete the cognitive task and think about, is estrogen pollution an issue? If you think it is, what should we do about it, or why isn't anything done about it? Because there's proven scientific studies that prove that it has an impact, but is that impact an issue? It's up to you. If you don't think it's an issue, you need to say why not. So read through that on Cognitive, please.